Cells are the fundamental unit of life. So they are the fundamental unit by which we understand how living things work. The smallest living things are cells. What I'm relating to you is what's called cell theory, really. So cells were discovered first by an Englishman named Robert Hooke, who also helped to develop some of the first microscopes. And once you could see things smaller than you could with the naked eye, it opened up a whole new world of biology. And he named these things cells. Didn't know exactly what he was looking at, but since then we've come up with quite a few theories about cells. And the first one is the cell theory. Okay, so Schleiden and Schwann were the two scientists who developed this theory. And it is this. First, that all organisms are made of cells. So stuff that I already mentioned before. And the processes of metabolism and inheritance occur in the cells. And we know this to be true based on DNA and what we know about that. Cells are also the smallest living things. So if it's smaller than a cell, like a virus, or a virus has a number of other characteristics which make it not a cell. But these are the smallest living things that we consider to be alive. Cells come from the division of other cells. So you can't build cells from nothing. Well, it had to have started the first cell that way. But ever since then, now we have cells which are building on top, off of each other. So cells are restricted in their size by the rate of diffusion. We'll talk about diffusion later. But essentially, diffusion is the way substances move naturally. And some substances need to go in and out of the cell. So, for example, most cells require oxygen, and so let's say this little red with blue border oval here is oxygen. If every part of the cell needs oxygen, then the cell itself can only be so big before oxygen can't get to those central parts of the cell because the distance is too far to get the oxygen to where it needs to be. Whereas in a smaller cell, cell 2 here, it's going to be able to diffuse at a rate which is fast enough to get oxygen to every part of the cell. So this really is a function of surface area to volume ratio. Other things that are going to affect diffusion, however, like I said, surface area, the temperature, the concentration gradient of whatever substance is going in and out, and then of course the distance. Okay, so this is really just a figure from your book explaining the relationship between surface area and volume. In bigger things, they do not have as much surface area compared to how much volume is inside them. In smaller things, they have a lot more surface area compared to the volume within them. Okay, and that affects how big a cell can be. Microscopes, like I said before, were fundamental to being able to look at cells and study cells. It increased our ability to see two objects as separate, okay, which is resolution. A compound light microscope has a resolution of 200 nanometers. A nanometer is 1 to the 10 to the negative 9th meters. Electron microscopes are able to have a resolution of 2 nanometers, so 100 times stronger than the light microscopes. These are big and expensive, and we don't have access to them, but they make a lot of really cool images, like the ones to the right here. So a scanning electron microscope is generally able to make very defined images or see things which are very refined on the surface, whereas a transmission electron microscope can actually see things that are within the cells themselves. Transmission electron microscope beams electrons through the material. The scanning one, of course, does it on the surface. So what have we learned about cells? First of all, we can generalize a few characteristics about all cells. All cells have DNA. That's part of the inheritance, which we talked about before. All cells have a concentrated area where the DNA is, either the nucleoid or the nucleus. All cells have ribosomes. Ribosomes are necessary for making proteins, which are the functional part of the cell, so you have to have ribosomes. And a cytoplasm. So this is a solution of the necessary compounds and minerals and macromolecules that are needed for life. And it's on the interior of the cell. And lastly, a plasma membrane, so something holding it all together. Okay, and we'll talk more about the plasma membrane and the structural components later. So we have prokaryotes and eukaryotes. These are the two types of cells. Prokaryotes are single-celled. These are generally smaller than our eukaryote organisms. And every organism is just this one cell. They do not have a nucleus. Instead, they have this nucleoid, which is a concentration of DNA of the chromosomes in the cell. The cell wall in prokaryotes is made of a structure called peptidoglycan. This will come up again and again when we talk about different organisms. Peptidoglycan in the bacterial cell wall. This is important for protection. It is different, however, than the cell wall in plants and fungi. It's made of a different substance. The prokaryotes do not have organelles. Organelles means little organs, are specialized functional parts of the cell. So the prokaryote cell is simpler. Some of them do contain accessory structures such as flagella and cilia. And those are generally for movement. There are two domains. So the domains are the major classification groups of living things. So there are three of them. 2 out of the 3, archaea and bacteria, are prokaryotic. Eukaryotes have a membrane-bound nucleus. And you see the cell itself is a lot more complicated together when you put everything together. It has membrane-bound organelles as well. It has a cytoskeleton, and it has an endomembrane system. So the endomembrane system, you can see this highlighted in the picture to the bottom right. It has a number of membranes, phospholipid bilayers, which form compartments which package and repackage things and send them around the cell. All right, so we're going to go through these organelles of the eukaryotic cell. First is the nucleus. This is the most simple. It's the part that holds the DNA. In prokaryotes, you have circular DNA. In eukaryotes, you have linear structures called chromosomes. And a chromosome is protein surrounding the DNA and packing it together. The nucleolus is the place where ribosomes are synthesized, and it's generally a darker spot in the nucleus. And it's surrounded by a lipid bilayer, actually 2 lipid bilayers, which is called the nuclear envelope. And in there are proteins which make pores, which allow things to go in and out of the nucleus. Ribosomes, these are not membrane-bound organelles, but they are an important part of all cells. They are made of ribosomal RNA, which we mentioned before. We talked about macromolecules, and their purpose is to make proteins or to synthesize proteins. There are 2 different types based on their location. So free ribosomes are in the cytoplasm, just floating around. And then you have ribosomes which are on the rough endoplasmic reticulum, so bound ribosomes. The endoplasmic reticulum then is one of the members of the endomembrane system. You have 2 different types. Rough ER has ribosomes embedded within it, and this allows the ribosomes to make a protein, and then the endoplasmic reticulum can then package it with a membrane and send it somewhere else. Smooth endoplasmic reticulum doesn't have the ribosomes on it, but is still involved in packaging, accepting these packages, and making modifications to things as they are sent throughout them. It also can be for storage, such as calcium, and is an important component in lipid and carb synthesis and modification, and can also help detoxify certain substances. All right, the Golgi apparatus is similar to the smooth ER. It's also in this network of endomembrane network. It's composed of flattened stacks of interconnected membranes, and its principal purpose is to transport molecules within the cell or outside the cell in structures called vesicles. Okay. It has two ends. The cis end is where things come into the Golgi apparatus. The trans is where things are sent out of the Golgi apparatus. It's sometimes called Golgi bodies. All right, one of the things that can be formed through the endomembrane system is a lysosome. A lysosome is a vesicle that has a bunch of enzymes in it, which can be used to break down old organelles, damaged organelles, food. Basically, those enzymes just help break it up into smaller pieces, and then it can be recycled and made into new proteins or whatever it needs. Another little lysosome with something in it is called a peroxisome. These have enzymes for converting hydrogen peroxide into water. Hydrogen peroxide is reactive and can destroy different cellular components. So it's needed then to contain and detoxify hydrogen peroxide. It also contains enzymes that oxidate fatty acids, which is important for breaking them apart and to be used for energy. All right, vacuoles. Vacuoles aren't found in all eukaryotes. They are found in most, if not all, plants. It's a membrane-bound structure, and depending on the cell, can have different function. A central vacuole helps to keep the plant shape constant or consistent, or vary depending on other factors. There are also storage vacuoles, which will store molecules for when they are needed. Some fungi and protists also have contractile vacuoles that may help propel water or fluid in or out of the cell. All right, mitochondria. Mitochondria are the powerhouse of the cell. This is where all the energy is being made. And the process of making energy is through oxidative phosphorylation. It also has a double membrane similar to the nucleus. And the inner membrane is folded to form these structures called cristae. They'll kind of look like little fingers in foldings. They have their own DNA. And so they can actually reproduce themselves, and their DNA is important for their individual function. You then inherit your DNA from your mother, not your DNA, your mitochondria from your mother. So in the process of sexual reproduction, females actually contribute more, obviously, but even in the beginning parts of the growing fertilized egg, the female is contributing mitochondria in addition to all the other nutrients. All right, chloroplast. This is another organelle with a double membrane, and it is the site of photosynthesis in plants. It can photosynthesize because it has this pigment called chlorophyll, which can be excited by sunlight, and it can excite electrons, and then those electrons can be used for energy. It also has its own DNA, and it has an inner membrane, which you can see at the bottom of this picture, and that inner membrane forms these stacks called thylakoid. All right, so there is a theory then for some organelles as to how they formed inside of an eukaryote. And this is called endosymbiosis. The theory is that chloroplasts and mitochondria originally were individual prokaryotic cells that somehow got engulfed by a larger cell, and the relationship became mutually beneficial, so that both were gaining in being closely associated with, in so much that they created a more efficient cell. And the evidence for this include that both have double membranes. So the idea is that this precursor cell was engulfed, and now it had its own membrane with the membrane around it. They also have their own DNA. Okay, so why would it have its own DNA if it was a part of the original cell? And they also have their own ribosomes. So there's evidence that they were once individually existent. But now they become so dependent on each other that you can't take the mitochondria or the chloroplast out and the cell can continue to live, either cell. Okay, so we call this mutually beneficial relationship a symbiotic relationship, one of a few different types, which occurred through different prokaryotic organisms. All right, eukaryotes also have a cytoskeleton, which is a network of proteins similar to your skeleton, but rather than made out of bone, it's made out of these proteins. This helps the cell maintain its shape. It also gives the cell, tracks and structure so that they can move things throughout the cell. So it helps in transport. And then it, generally just holds things in place. The three different types of proteins are microfilaments, which are the smallest, microtubules, which are the largest, and then intermediate filaments, which are between the two. Okay, and this is from your book. You see examples of each. All right, some things in the cell do not have an active function all the time. So for example, centrosomes, these have microtubules within them that help to guide cells and chromosomes during cell division. They're also a principal component of flagella and cilia. But plants and fungi lack centrioles, so they have actually a different method for dividing the chromosomes for cellular reproduction. Cell walls, these are also not found on all cells, but plants, fungi, and some protists have a cell wall. It's an extra layer of protection. In plants and protists, this cell wall is made of cellulose, which is a carbohydrate we talked about. In fungi, it's made of a different carbohydrate called chitin. And so those are the organelles. There are, however, many other important components of a cell which help in its function. One of them is the extracellular matrix. So especially in multicellular organisms, there are a lot of things occurring outside the cell which help coordinate with other cells. It's often made of glycoproteins. So glyco refers to sugar molecules, protein, of course, to protein, or collagen, which is a protein, or both, in these multicellular organisms. This performs a protective layer outside the cell, so it keeps things from getting in that shouldn't be in there. All right, so here's a figure. This is not from your book, it's from a different book, but just showing the similarities and differences between animal and plant cell and prokaryotic cells. Looking at the different organelles, membrane-bound organelles that prokaryotes do not have, and other differences between the two. And I circled and highlighted some of the important parts. We'll go over this in class as well. Okay, so many cells, especially in a multicellular organism, need to communicate with each other in order to function. So in order to do that, they first have to recognize each other as, from the same cell or a different organism altogether. So they have recognition properties. This is done through surface proteins, including glycolipids. Glyco means sugar, lipid means, of course, lipid. So this is a combo macromolecule. And then there's also MHC proteins, matrix histocompatibility proteins. These are used by the immune system to recognize them as foreign or not foreign, as okay. You also have in multicellular organisms the ability to tie cells together. Okay, so you may have a barrier of cells, but they need to be tied together to make a complete barrier, and this is done through a connection called a tight junction. Okay, this form a waterproof barrier. Sometimes they allow some water through, or some types of molecules and not others. Desmosomes. This is more structural rather than a barrier. So if you have two cells which are experiencing constant force and trauma, they may need a little extra support to be held together. So an example of this might be heart cells. They have a lot of desmosomes because they're constantly moving. All right, and then you have gap junctions and plasmodesmata. These then allow signaling molecules to pass through from one cell to the other, and thus allow the cell to communicate with the other. Okay, and here's a picture of a plasmodesmata showing a connection through the cell wall to different types, two different cells. All right, so that's kind of the general parts of the cell. Now we're going to talk about cell membranes and signaling. So how we talked a little bit about signaling, but a little bit more about how, especially in multicellular organisms, they're able to communicate. So first off, membrane structure. Okay, so the fluid mosaic model is the theory for how a lipid membrane is organized. So the idea of fluid, that means is referring to the lipid bilayer as not being static, not being just one concrete collection of molecules and compounds, but is actually something that is constantly moving and changing. Okay, and a mosaic meaning there are lots of different things embedded within there. However, the principal component of it is these phospholipids, which we talked about with macromolecules. These have hydrophobic and hydrophilic ends. Because most cells are in water, the hydrophobic ends are towards each other on the inside, and the hydrophilic ends are towards the outside. Within there, you also have proteins, so embedded. So here I have a picture of a little figure of a little protein. It may be embedded on the inside, outside, or across the lipid bilayer. And there's another structural component, cholesterol, which helps to break up if your phospholipids have saturated fats which pack tightly together. The cholesterol can help break that apart a little bit. All right, these bilayers form spontaneously in water, and we went over this. It's because they have the hydrophobic end and the hydrophilic end, which are attracted and repelled with water. They can also be organized into functional sections. So one example is a lipid raft, which is a formation of cholesterol and a specific type of phospholipid called a sphingolipid. Okay, these can form in the endoplasmic reticulum, where you're making these lipid bilayers and then sorted out in the Golgi apparatus into these specific arrangements of lipids and cholesterol and whatever may be in them. All right, we went over the structure, so I'm just going to briefly go over this. You have the choline, which is the top part, the phosphate group, the glycerol, and the 2 fatty acids attached. That is what are the components of a phospholipid. Now, the proteins then embedded within them, they have lots of different functions. So I said they could be in or outside or across. An integral plasma membrane protein is a protein that is embedded within the plasma membrane. They can be important for transport, channels, attachment, recognition, receptors, enzymes. So they have lots of different functions, just like when we talked about the many different functions of proteins. There are also peripheral plasma membrane proteins. So these are on the outside or the inside. They are important for cell and tissue recognition, such as with glycoproteins and glycolipids. They are shuttle carriers, where they may take things across the lipid membrane. And they're also important for the interior protein network, which helps to maintain the shape, anchor the proteins, and combine with specific molecules. So you can see all of those on this figure here from your book. All right, cholesterol we mentioned before and how it was important to help break up this fatty acid structure. And it increases permeability. And it can do that because it also has a hydrophobic and hydrophilic structure to it. It has a polar head on it, which is going to be hydrophilic, and that's going to point towards, of course, the water, the outside or the inside of the lipid membrane. And then it has a lipid structure and a hydrocarbon tail, which fits nicely into the lipid part of the phospholipid bilayer. All right. Finally, just wanted to reiterate this concept in biology that the structure relates to the function. Okay, so diverse functions arise from the different structures of these membrane proteins. So, for example, in order to be an integral plasma membrane protein, a protein has to have these nonpolar sections, which can fit inside the lipid part of the phospholipid bilayer, and these hydrophilic ends, which can point outside and inside. These structural components can be generalized, and common features can be attributed to those functions. And some of these include, which are important to their function, include nonpolar portions, like I mentioned before, chemical bonds made within them, transmembrane domains, and making pores in the membrane as well. All right, so now we're going to switch. We're going to talk about permeability. So this is the ability of substance to pass through another substance. So we're going to talk about the permeability of the cells, which is due to these proteins and structure of the phospholipid bilayer. Okay, so now so the lipid bilayer is a semi-permeable membrane, meaning it lets some things through and it keeps other things out. Okay, and it needs to be this way to allow the things that you need in, to release waste, to keep other things out. Molecules pass through the membrane via 2 methods, which are really general methods for how molecules move generally, even without, even not through the membrane. And these are passive and active transport. Passive transport occurs naturally without any force involved, without any energy. Active transport requires the use of energy, and the molecule in living things which represents energy is ATP. So that's usable energy in cells. All right, passive transport, like we said, doesn't require ATP. It is the molecules flowing from greater to lesser concentrations. So you have this little figure here. You have a concentration of this green substance, these green dots in one end, and a low concentration in the other. So naturally, these green dots will flow to the right. Okay, diffusion is simply that. Osmosis is another process dealing with water. Facilitated diffusion is diffusion through a channel, and filtration or bulk flow is due to pressure. So we'll go through these individually. So first, in a solution such as a liquid or a gas, molecules are going to flow from greater to lower concentration until they are all evenly spaced out. That's diffusion. In a solution, you have the solvent, which is usually water, and the solute, which are the molecules within them. We've been talking a lot about solutions in lab, so this should be familiar to you. One example is a drop of dye in water. The dye is the solute. The water is the solvent. Okay, so that's simple. You have a lot of concentration of something in one area. It's going to even out throughout the whole area. Osmosis then is like diffusion, except it's through a semi-permeable membrane and the water is flowing, not the solutes. So if you have two solutions separated by the semi-permeable membrane that will not let the solutes through, but will let the water through, the water will go through the membrane to even out the concentration of the solutes. Okay, so you see that here. You have a membrane. There's a lot more of the green substance on the right. And we have the sugar molecule as the green substance. And you have the semi-permeable membrane down the middle. What the water is going to do is going to try and even things out by passing through the semi-permeable membrane. And it'll continue to do that until it has a concentration on both sides, which is equal. So when we're talking about solutions and different concentrations of solutes, you can talk about them in relative terms. So hypertonic is the solution that has the greater concentration of solutes. Hypotonic is the solution with the lesser concentration. And isotonic is when they are in equilibrium and they have the same number of solutes in their solvent. So osmotic pressure then is the force needed to stop this osmotic flow. So you can see this in a cell. A cell in a hypotonic solution is going to gain water, causing it to swell, and this is going to create then pressure. If the membrane is strong enough, it'll reach a counterbalance between the water coming in and the strength of the membrane. But if it's not supported by something like a cell wall, such as in plants and fungi, then it may burst. So you see that to the left here. Okay, if the opposite is true, if water is flowing out, then the cell may shrivel up if it doesn't have enough force to counteract that balance. All right, facilitated diffusion is very simple. It's just diffusion of a substance, but through some sort of protein that it assists it. Without the protein, it couldn't fit through the lipid bilayer. So these can either be protein channels or protein carriers. They don't require ATP to allow the things pass through. So again, it's passive transport. These proteins can be closed or open so that they can be selective. And an example is aquaporins. So aquaporins are channels that allow water through. If you don't want water to come through, then you can just close the aquaporin. All right, so channel proteins can allow different things in, such as charged particles, and they can be gated, which will open and close when stimulated. And the flow of ions is determined by the concentration gradient of the ions, the charge, and whether the gates are open or closed. All right, carrier proteins then can transport molecules such as glucose in and out of the cell. What happens with the carrier protein is it's open, the glucose goes in there and binds to it, and then it opens on the other end, allowing it to go through. For each of these, there is a saturation point, a point at which the concentration of the solute will no longer allow a greater rate to pass through. So there's a maximum in how fast they can work. All right, finally, filtration or bulk flow. This is where a solution is forced through pores due to pressure. So an example of this is fluid passes through your blood vessels into your kidney, which is occurring in this figure. The force occurs because your heart is beating, but that's far upstream, and that's going to create then a pressure which forces water out. So at the site of filtration, there is no ATP needed because you have pressure already there. All right, the second half then is active transport, which requires ATP, and it requires ATP because it's against the natural flow. So in this, we're going to build a concentration. We're going to add more of a substance from an area that has less of the substance, so moving from low to high concentration. This requires selective carrier proteins and, of course, ATP. Some of the carrier proteins can be uniporters. Uniporters are going to move one molecule across at a time. So here you have a proton pump. It's going to move protons across the lipid bilayer using ATP. Symporters move two molecules at the same time. So here we have diffusion of hydrogen passing through this sucrose transporter. As it passes through, it also grabs a molecule of sucrose, and they can go into on the other side of the lipid membrane. Antiporters, they aren't pictured here, but are similar to symporters, but they move in opposite directions. So if the sucrose was moving from left to right rather than right to left, while the protons are moving from right to left, that would be an antiporter. These can also be used to describe facilitated diffusion carriers. All right, bulk transport then is using membranes to transport cells in and out of the cell. So packaging something and then moving it inside or outside. Endocytosis means moving something into the cell. So some examples are phagocytosis or pinocytosis. Okay, phagocytosis is moving food or some sort of energy molecule. Pinocytosis is moving water inside the cell or some sort of solution. Exocytosis then is the opposite, and we don't have that pictured here, but that's moving things out of the cell using the membrane. And this, because it's an active transport, is something that requires energy. Here's an example of exocytosis. You may have a secretory vessel, something you want to secrete to the outside of the cell. It will then bind with the outer plasma membrane. As it binds, whatever's inside of it is then excreted out. All right, finally, last little section we're going to talk about is cell signaling. So when cells are far away from each other, they may use chemicals to invoke changes in other cells. The chemical that is going to invoke that change is called a ligand, or that's the signaling molecule. It oftentimes doesn't work by itself and requires a receptor to bind to, which is on the target cell. You can have an extracellular then binding, or you can have an intracellular binding of the inside the cell inside. So signals from one cell to the other can occur across different levels. In an autocrine signal, that's going actually within the same cell, from one part of the cell to the other. In a paracrine signal, it's going from the same tissue. So it's from one cell to the other, but it's within the same general area, because tissue is an aggregation of cells that have the same function. Endocrine signals, these are from far away. So this is from a cell from one tissue to another cell in a different tissue. Okay, and these usually travel through blood. So these are also called hormones. And then finally you have synaptic signaling, which occurs from nervous tissue. All right, so these are different types of signals. Here's the figure from your book showing the different types. Okay, and that's it for cells and cell signaling.